Hi and welcome to this video. So this is Miss Pedral and I'm gonna be discussing about cell cycle and cell division. Now you're probably listening to this video because we weren't able to finish our discussion inside the classroom due to time constraints. So by the way, this video is actually included in the midterm. So if I were you, I'll take down notes and pause it from time to time to absorb the information. And you can actually repeat the video if you want to. Okay, now if you have any questions, feel free to ask me or just send me a message. Now, for the cell cycle and cell division part, when we say cell cycle, this one actually pertains to a sequence of development and division that all cells go through. Now, it doesn't matter if this type of cell is a body cell or a sex cell or your gametes, as long as it's a cell. Okay, once in their lifetime they have actually went through the process called the cell cycle. Now, why is the cell cycle necessary? Well, first, for the continuity of life, of course, and then second is for growth, repair, and of course development. Like for example, if you got wounded, after some time, those wounds will heal, right? Now, probably the reason, or not probably, but simply because of cell cycle and cell division. So, that's one of the reason why we need cell cycle in our lives. Now, cell cycle is actually divided into the following: the interphase, cell division proper, which includes mitosis and meiosis, and of course, we have cytokinesis. Now, we will try to identify the difference between the three as we go along the discussion. And let's start. For the interface, we refer to this one as the non-dividing stage simply because the cell, whenever they are in interface, they don't do any types of division. What they do is they replicate their materials, or they replicate the material so that upon cell division, the materials that they have will be divided into two equal parts. So both of the cells that are the products of your cell division will have the same genetic material as the other one. And then since cells devote their time largely to cell growth, this is also the part where they are metabolically active. Now, when we say metabolically active, it's the time wherein they made use of the energy from the food for growth and development. And then, interphase is also the longest stage in the cell cycle. Now, interphase is divided into the following phases. First, we have G1 or the gap 1 phase. Now, during gap 1, the cell increases in size in preparation for cell division. And just like what I told you a while ago, cells actually made use of their time here to increase in size, to grow, and of course, to replicate their materials. Now, take note that in gap 1 or in G1 phase, the only things or the only parts being replicated here are your organelles and the different structures inside the cell. So for G1, take note that the replication does not include the replication of the genetic material, only the organelles and your structures. Okay, why? Because there's another part in your interface where the DNA are replicated. And then we have G0. Now, G0 is a checkpoint. Okay, please bear in mind that while at G1, some cells enter the G0 phase or the quiescent stage. Now, at G0, cells are not able to move to the S phase and thus are unable to replicate their DNA. Now, some cells enter G0 because of three reasons. First, because they don't have enough nutrients to continue to the next phases. That's why they stop here. Second is because there are errors, or they are checking, the cell is checking for possible errors made at the gap 1 phase. And they enter G0 so that those errors will be corrected. And then lastly, cells enter G0 because they are already matured. Like for examples, your heart muscles, your eye cells or the cells inside your eyes, and of course your brain cells or your neurons. So it doesn't mean that when they enter G0 they don't have enough nutrients. It's just that sometimes at some point in their lives they already reach the peak or they are already matured or differentiated. Okay. Next we have the S phase. So S phase, this is now where your DNA is being replicated. So genetic material and chromosomes are duplicated in your synthesis phase or the S phase. Next, we have the G2 phase or the gap 2. Now, for the gap 2, di ba, a while ago, G0 is the checkpoint for G1. Now, for S phase, the checkpoint is actually the G2 phase. So, the cell double checks if the chromosomes are synthesized and replicated properly. Now, if there are any errors, if there are any letters, your ATCG are not paired properly, then this is where the time that the cells will fix them. Okay? Also, aside from that, proteins are being synthesized and the cell continue to increase in size in the G2 phase.